Hello, my name is Anastasia Chapko, and I am a computer engineering and neuroscience double major at the University of Cincinnati. I regularly volunteer with Coach T's nonprofit called Nerds, and I am an undergraduate research assistant at Dr. Jha's Mind Laboratory. I will also be the presenter for this RET project's Neuroscience Background videos, which is the first of. So let's dive in. First off, what is neuroscience? Neuroscience is a scientific study of the nervous system, and our nervous system is subdivided into two main parts: the central nervous system, or CNS, and the peripheral nervous system, or PNS. The CNS consists of our brain and spine, while the PNS includes all the nerves which branch out to our limbs and organs in our body. So why should we care about the study of our nervous system? Or in other words, why is neuroscience important? Neuroscience is important in part because of all the things it can tell us. Our brains are responsible for who we are, for our behaviors, our emotions, our thoughts, and our consciousness. Our brain is our central processing unit, our CPU. So by studying neuroscience and understanding more about our brains, we are really understanding more about ourselves. Not to mention, since everything we experience goes through our brain, everything gets filtered through whatever architecture the brain has. Understanding that architecture helps us understand the outside world better, as well as see what biases we might be applying to everything that we experience. Neuroscience is also important because we can do a lot with it. There are over 1,000 disorders that affect our nervous system and brain. Alzheimer's, Parkinson's, epilepsy, and autism, and many others are examples of such disorders. A logical application of neuroscience comes in the form of studying, understanding, and developing treatments or cures for diseases and disorders like these, which affect our nervous systems. Another potential use has surprisingly little to do with biological applications. Our brain is an amazing computer, which can do a lot of things that our actual technological computers cannot, or at least could not in the past. Technology has taken note and inspiration from the brain to create groundbreaking artificial intelligence, not so loosely based on the mechanisms at work in our own heads. Thanks to the understanding we've gathered about how our brains work, we were able to apply the same concepts to computers and created artificial neural networks, which give us our voice assistants, which rely on natural language processing, our automated data analysis softwares, self-driving cars, facial recognition, and a bunch of other really cool technology. Now that we know some of the reasons why neuroscience is important, let's look at what we're going to cover in this RET Neuroscience module today. For the purposes of our modules, we will focus on the brain, which is part of our central nervous system, or CNS. In the following sections, we will cover neurons, action potentials, synapses, and neural networks. So let's begin with our first topic, neurons. What are neurons? Neurons are cells of the nervous system, so they are also called nerve cells. Neurons receive, transform, and transmit electrical signals. And they can also interface with many other neurons to form neural networks, or neural circuits. So we now loosely understand what a neuron does, but what is it made up of? What are the parts of a neuron? Neurons will typically have three parts: the dendrites, the soma, and the axon. Dendrites are the inputs into a neuron. They receive signals from the outside world or from other neurons and send them to the cell body, or soma. The soma processes those incoming signals and decides whether or not to pass the signal along. The axon serves as the output of a neuron. The signals generated by the soma flow down the axon to whatever is connected downstream. And just as a note, not all neurons have just one of each of these items, and not all neurons look like the simplified image on the slide. Neurons vary in size, shape, and function greatly, as shown in this next image. As you can see, there are many different architectures a neuron can have, so just keep that in mind for the future. So we've been talking a lot about signals coming into cells, being processed by cells, and being sent out to other cells. But what exactly are these signals? They are action potentials, which are spikes of electrical activity. The chart on this slide shows voltage over time. The line starts off flat, then spikes sharply up, followed by a sharp trend downward, and then levels out to horizontal again. That spike, that up and down motion on the chart, is an action potential. So let's dig in a little more. There's something called a membrane potential. The membrane potential is the voltage that we can measure from the membrane of a neuron, or nerve cell. When the cell is at rest and nothing too exciting is happening, the membrane potential will typically be at negative 70 millivolts, and this value is called the resting potential. So let's think of an example neuron. This neuron's membrane is at the resting potential of negative 70 millivolts. As action potentials from upstream neurons reach the dendrites or inputs of our example neuron, the membrane potential of our example neuron depolarizes or goes up. So it started at negative 70 millivolts, now it might be at negative 65 or negative 60 millivolts. Whatever it may be, it's just inching up closer to zero and positive values. At some point after enough activity, the example neuron's membrane will reach something called the threshold voltage. This is the voltage needed for an action potential or spike to fire. Once the membrane potential reaches the threshold voltage, the cell quickly depolarizes or becomes more positive, and then hyperpolarizes or becomes more negative. That right there is, again, the spike or action potential. So one more important little factoid. Action potentials are a fixed size. They always have the same height and shape for each cell, but also there is no such thing as a partial action potential. It either happens, it fires, or it doesn't. There is no in between. Now we've talked about the signal that neurons receive, process, and send out, but we still haven't talked about the connection between neurons through which the signal gets passed on, the synapse. What are synapses? Synapses are the connections between neurons. They are tiny gaps across which our signals, or action potentials, pass through on their way from one neuron to the next. These connections are usually between axons, or outputs of one neuron, and dendrites, or inputs of other neurons. But this is not always the case. For example, there are sometimes connections between dendrites and dendrites, or, less rarely, axons and somas. But during these RET modules, we will assume the typical case of synapses forming between axons and dendrites. Let's consider one of these typical synapses as an example. If you zoom in on this example synapse, you will see a presynaptic terminal, a postsynaptic terminal, and a gap in the middle. The presynaptic terminal is the terminal before the synapse. In other words, for our example synapse, it's the part of the axon that is touching the gap near the dendrites of the neuron it's typically sending a signal to. The postsynaptic terminal is the terminal after the synapse. In other words, again, for our example synapse, it's the part of the dendrite that is touching the gap near the aforementioned axon of the neuron that it's typically receiving the signal from. Now that we understand a little about what a synapse is, we must look at a further distinction. There are two different types of synapses: chemical, which are more complex but slower, and electrical, which are less complex but faster. Most of our synapses are chemical synapses, since they are more flexible and can achieve different outcomes. Chemical synapses use chemical messengers, called neurotransmitters, to transmit information between neurons. And some of these neurotransmitters, even if they may seem like a relatively foreign concept, are actually well known in our day-to-day life. Adrenaline, dopamine, and endorphins are all examples of neurotransmitters. Let's try to get an idea of how an action potential would look from the perspective of our chemical synapse. An action potential that is fired from the presynaptic neuron triggers neurotransmitters to be released from the presynaptic terminal. These neurotransmitters then diffuse across the gap between the pre- and postsynaptic terminals and bind to their associated receptors on the postsynaptic terminal. These bindings then cause other events to happen, which will result in the postsynaptic cell either depolarizing, having its memory potential go up, or hyperpolarizing, having its memory potential go down. These events depend on the neurotransmitters and receptors involved. Because of this mechanism, where neurotransmitters can only be released from the presynaptic terminal, and the receptors which cause the events necessary for depolarization and hyperpolarization are only on the postsynaptic terminal, the flow of our signals, our action potentials, always goes in one direction, from presynaptic terminal, the axon of the previous neuron, to postsynaptic terminal, the dendrite of the following neuron. This one-directional flow is not so for electrical synapses. Electrical synapses have a physical connection in the gap between the pre- and postsynaptic terminals. These physical connections are called gap junctions, and they allow electrical current, or ions, to flow through them. Because these gap junctions are just channels for ions to flow through, there is no restriction on which direction the signal gets passed in. This means a presynaptic neuron firing can cause a depolarization in a postsynaptic neuron, like normal, but also that a postsynaptic neuron firing can cause a depolarization in a presynaptic neuron. Now we know a bit about chemical and electrical synapses. We're almost done here. We've talked about what neurons are made of, the signals they process, and the way they can connect to each other. We just need to cover what happens when they do connect to each other, when they form biological neural networks or circuits. A biological neural network or neural circuit is simply a network made up of neurons that serves a specific function when it is activated. These networks are formed through many neurons having many synaptic connections with many other neurons. And it's important to note that one neuron is almost always a part of multiple neural networks, which is not surprising considering that the average neuron has about 1,000 synapses, or 1,000 connections to other neurons. These different networks of interconnected neurons are what make up our nervous system and are responsible for all our thoughts, behaviors, ideas, and everything that makes us, us. So let's finish things up. In this video, we have covered the following questions. What is neuroscience and why is it important? What are neurons and what are their parts? What are action potentials? What are synapses? And what are neural networks? With this information, you should be ready to move on to the next modules on bio-inspired computing, electronics, and the spiking device. Thanks for watching and enjoy the rest of the modules.